Hello student. Now we are going to discuss another topic in respiratory physiology, that is carbon dioxide transport. So how carbon dioxide is transported in venous blood. So in this session what we are going to discuss, first of all we are going to discuss various methods of carbon dioxide transport in venous blood. Then we are going to discuss two important things, that is one is Bohr effect and second one is Haldane effect, and we are also going to discuss carbon dioxide dissociation curve. So in the previous chapter we have seen the oxygen dissociation curve. Here we are going to discuss the carbon dioxide dissociation curve, and at the end of the session we are going to discuss few MCQ. So this is our plan. Now let's start with the first topic, that is methods of carbon dioxide transport. So look at this schematic diagram. We have seen this schematic diagram earlier also. So this is the representation of the lung, or you can say this is the representation of the alveoli. So this is the representation of the lungs. So at the level of alveoli, if I ask you what is the partial pressure of carbon dioxide, your answer will be 40 millimeter of mercury, right? Now this is the circulatory system. So this red color diagram, and this side on my right hand side, this is the arterial system. Okay, so this is the arterial system, and on other side this is the venous system, right? So this is the venous blood where I am putting this arrow. Okay, now if I ask you that inside the arterial system, what is the partial pressure of carbon dioxide in the arterial blood? Again your same answer, that is 40 millimeter of mercury. But whenever I will ask you that what is the carbon dioxide content in the arterial blood, so carbon dioxide volume, okay, so that is 48 ml per deciliter of blood. So content or amount of the carbon dioxide which is present in the arterial blood is 48 ml per deciliter of blood. Okay, now when this blood is going at the level of the tissue, so we all know that from this arterial blood oxygen will be taken up by the tissue, and the tissue is going to produce carbon dioxide. So the tissue is going to produce carbon dioxide, right? So for 100 ml of blood, okay, how much carbon dioxide will be added up by the tissue? So the carbon dioxide production is 4 ml per deciliter of blood, right? So whenever this 4 ml of carbon dioxide is added with this 48 ml of carbon dioxide, what will be the carbon dioxide content in the venous blood? 52 ml per deciliter. And if I ask you what is the partial pressure of carbon dioxide in the venous blood, your answer will be 46 millimeter of mercury, or some of the books are saying 45 millimeter of mercury. So this is the overview of the carbon dioxide transport. So here what we are saying, that we are saying that the difference between carbon dioxide content of the arterial blood and venous blood is 4 ml per deciliter. So 4 ml carbon dioxide, where from it is coming? It is coming from the tissue level. Tissue is producing continuous carbon dioxide because of the metabolism. And because of this extra amount of carbon dioxide is adding up in the venous blood, the partial pressure of carbon dioxide is also raised in the venous blood, that is 46 or 45, and in arterial blood this is 40. So in carbon dioxide transport, we are basically going to understand that how this extra 4 ml of carbon dioxide is carried by this venous blood. That is the topic, that how this extra 4 ml of carbon dioxide is carried by venous blood. So now let's look at the various methods of carbon dioxide transport in venous blood. So venous blood transport carbon dioxide with the help of the plasma also and the RBC also, means RBC also transport carbon dioxide and plasma also helps in carbon dioxide transport. And in both the cases, within the plasma also and within the RBC also, there are three forms by which carbon dioxide is transported in blood. So plasma, inside the plasma there will be certain amount of dissolved carbon dioxide, carbamino compound formation and bicarbonate. So carbon dioxide is transported inside the plasma by this three format. And in the same way, the carbon dioxide is also transported inside the RBC by the formation of the dissolved carbon dioxide, by the formation of carbamino compound, by the formation of bicarbonate. Now let's look at, so dissolved carbon dioxide, it is 5% of the total carbon dioxide transport in case of plasma also as well as RBC also. So dissolved carbon dioxide means this is the carbon dioxide which is present inside the water portion of the plasma and water portion of the RBC, that is the dissolved portion, okay, dissolved in the plasma, dissolved in the water, right? Now whenever I will say carbamino compound, whether it is plasma or RBC, what is the meaning of carbamino compound? Carbamino compound means if the carbon dioxide is attached with the amino group of protein or maybe hemoglobin. So amino group, where it is present? Amino group is present inside the protein also. So inside the plasma there are plasma protein. So with that plasma protein amino group, when the carbon dioxide is attached, that carbon dioxide is known as the carbamino compound of the plasma. And inside the RBC, protein is not that much, but the carbon dioxide attached with the amine group of hemoglobin. So this is also the carbamino compound inside the RBC. Getting my point? Now if you look at the percentage, so this carbamino compound inside the plasma is only 1% of total carbon dioxide transport, but inside the RBC this is 21% of total carbon dioxide transport. So inside the RBC, the carbamino compound formation is very high in comparison to the plasma. Why so? So if I ask you that inside the plasma, carbamino compound is formed by which molecule? The protein molecule. Protein, the amine group of protein is combining with the carbon dioxide. And what is the concentration of protein in the plasma? We all know that the amount of protein or the concentration of protein in the plasma is 8 gram percentage. But if I ask you that what is the concentration of hemoglobin, what is the concentration of hemoglobin which is forming the carbamino compound inside the RBC? What is the concentration of hemoglobin inside the RBC? Then your answer will be 33%. So where from this 33% is coming? This is the hemoglobin concentration within the RBC. That is known as the MCHC, mean corpuscular hemoglobin concentration. What is the normal value of mean corpuscular hemoglobin concentration or MCHC? 33%. But what is the concentration of protein in the plasma? That is 8 gram percentage. So the molecule which is forming carbamino compound in the plasma is percentage is only 8 gram percentage. But the molecule which is forming the carbamino compound inside the RBC, that is 33%. So that's why the carbamino compound of the RBC is 21% and the carbamino compound of plasma is only 1%. Now the third format of the carbon dioxide transport as is written here, you can see here that bicarbonate formation, it occurs inside the plasma also, bicarbonate formation occurs inside the RBC also. But as you can find out here that the bicarbonate formation inside the RBC is very higher than that of the bicarbonate formation of the plasma. Why so? So we all know that the bicarbonate formation means carbon dioxide will react with water and this reaction will give rise to carbonic acid, and that carbonic acid will be converted into H plus ion or CO2 ion. Okay, so this carbonic acid will be converted into H plus ion and the bicarbonate ion. So that's why we saying that carbon dioxide is transported in the formation of bicarbonate ion. So this reaction require the help of an enzyme. That enzyme is known as the carbonic anhydrase enzyme. So this carbonic anhydrase enzyme which is present inside the RBC, this carbonic anhydrase enzyme, it is 5000 times faster than that of the plasma carbonic anhydrase. So because the carbonic anhydrase enzyme is 5000 times faster than that of the plasma, so obviously more of the bicarbonate is going to be formed inside the RBC rather than plasma. So that's why the percentage here is 63% and it is 5%. So now if I ask you maximum form of carbon dioxide transport in the blood, so blood means it is the combination of the plasma as well as RBC. So as you can find out here that carbon dioxide is mainly transported by bicarbonate formation. And how much is the percentage? 5% plus 63%. So on an average 68 to 70% of the total carbon dioxide is transported in the formation of bicarbonate inside the plasma and RBC. So this is the overall method, that what are the different methods of carbon dioxide transport, and bicarbonate is the main method. It has been asked multiple times in MCQ. Now after understanding this, now we are going to understand two important phenomena related to the carbon dioxide and oxygen transport. One is the Bohr effect and one is the Haldane effect. So this is also related to the carbon dioxide dissociation only, so try to understand. So Bohr effect, first thing if I ask you Bohr effect where it happens, your answer will be that Bohr effect occurs at the level of tissue. So the tissue level, okay, the Bohr effect is there. And the Haldane effect, okay, so I'll show you here. The Haldane effect, where it occurs, the Haldane effect occurs at the level of lung. So first you remember this, that the Bohr effect is at the level of tissue and Haldane effect is at the level of lung. Okay, Haldane, H-A-L-D-A-N-E, so H-A-L, L for lung. So that way remember. So Bohr effect at the level of tissue. So what is happening at the level of tissue? So we have already discussed that tissue means it is continuously producing carbon dioxide. So the carbon dioxide is produced inside the tissue, and the blood is coming to the tissue level. So this is the capillary through which the blood is coming. So this arrow indicates that the blood is coming from this direction, and the other arrow indicates that the blood is going out of the capillary through this direction. And inside this capillary, this is nothing but your RBC. So this structure is nothing but your RBC, a big diagram of RBC. So if I ask you that the blood which is reaching at the level of tissue here, which blood is coming, arterial blood or venous blood? Arterial blood. So this is oxygenated blood. So this is oxygenated blood. And oxygenated blood means hemoglobin that is reaching through this blood is HbO2, hemoglobin, oxygenated hemoglobin, HbO2. So oxygenated blood, and the hemoglobin that is reaching here inside the RBC, that is HbO2, oxygenated hemoglobin. So I am writing here that inside the RBC HbO2 is there. Now, whenever the carbon dioxide is formed at the level of tissue, because it's a gaseous molecule, so carbon dioxide will easily penetrate from the level of tissue and it will enter into the interstitial space. From there, the carbon dioxide will enter further into the plasma. The carbon dioxide is entering into the plasma. Now, whenever the carbon dioxide is entering into the plasma, it is going to generate three formats. What are the three formats? We have already discussed that certain part of this carbon dioxide will remain in the dissolved form inside the plasma. Certain part of this carbon dioxide will bind with the amino group of the protein. So that is a carbamino compound, and certain amount of carbon dioxide will form the bicarbonate. So these are the three formats by which carbon dioxide is transported in the plasma. But that is not the major portion. Major portion of the carbon dioxide will be transported by RBC. So this carbon dioxide which has entered into the plasma, now it is going to enter further inside the RBC. So now we look at the carbon dioxide which has entered inside the RBC. Again this carbon dioxide which has entered in the RBC, again this is going to form this dissolved format and carbamino compound. Okay, it is going to bind with the amine group carbamino compound. But these two are not the main. Which is the main? Main is the carbon dioxide is going to react with the water which is present inside the RBC, and because of this reaction, what is going to form? It is going to form the carbonic acid, and now this carbonic acid is going to divide it into H plus ion and the bicarbonate ion. Getting my point? Now this H plus ion which has just generated here, this H plus ion which has just generated here, what this H plus ion is going to do? This H plus ion is now going to react with the oxygenated hemoglobin which is present inside the RBC, HbO2. And because of this reaction, what is going to form? This reaction is going to form two products. One is the HHB, that is the deoxygenated hemoglobin, and it is going to release the oxygen. So look at this reaction. So oxygenated hemoglobin, that is HbO2, is reacting with the H plus ion. So it is forming HHB, that is the deoxygenated hemoglobin, and obviously oxygen is going to be released. So whenever this oxygen is released, now it will penetrate the membrane of the RBC. So it will reach into the plasma, and from that plasma, oxygen is going to be delivered at the level of tissue. So this way, tissue is getting oxygen by giving carbon dioxide to the RBC. Getting my point? Try to understand. So the tissue is producing carbon dioxide. This carbon dioxide is delivered inside the RBC, and because of this carbon dioxide delivery, the oxyhemoglobin which was here, this oxyhemoglobin is releasing its oxygen, and whenever the oxyhemoglobin is releasing its oxygen, what will happen? The oxygen will move from RBC into the plasma, and from there it will released into the tissue. So release of oxygen in presence of carbon dioxide, or you can say release of oxygen in presence of H plus ion. So ultimately carbon dioxide is generating this H plus ion, and that H plus ion is helping in the release of oxygen from hemoglobin. Getting my point? So this release of oxygen, okay, so this release of oxygen in presence of carbon dioxide or in presence of H plus ion, this is known as Bohr effect. This is known as Bohr effect. Getting my point? So now you understood Bohr effect. So Bohr effect means release of oxygen from hemoglobin. How or when it will be released? If the carbon dioxide is high, if the H plus ion is high. So carbon dioxide or H plus ion basically releasing the oxygen from hemoglobin. Which kind of hemoglobin oxygen dissociation curve shifting this is? This is right shifting of the curve. So that's why we also say that Bohr effect is the right shifting of the oxygen hemoglobin dissociation curve in presence of H plus ion or in presence of carbon dioxide. So if we look at book, so book is writing that when pH is lower, so acidic pH, H plus ion, or when the carbon dioxide is raised, the oxygen affinity of the blood decreases, oxygen affinity of the hemoglobin decreases, okay. That causes increased P50, that is the right shifting, leading to release of oxygen to the level of tissue. So this release of oxygen to the level of tissue in presence of increased carbon dioxide or decreased pH is the Bohr effect. The different definition is also there, that is Bohr effect is the right shifting of the oxygen hemoglobin dissociation curve caused by the concentration of carbon dioxide or pH. Getting my point? So this is also Bohr effect. Now look at, although we are saying that H plus ion or carbon dioxide, both of them are causing Bohr effect, but more it is because of mainly of the pH effect. So if you have to choose that Bohr effect is the effect of carbon dioxide increment or H plus ion increment, your answer will be that the effect of H plus ion, that is the pH buffer, is more stronger than that of the carbon dioxide. And the original Bohr effect was also discovered with the help of H plus ion only. So if you have to choose between carbon dioxide and pH, then you choose the pH for the Bohr effect. Getting my point? Okay, now we are coming back to the same diagram, original diagram. So now we are understanding that when carbon dioxide is entering inside the RBC also, it is forming dissolved carbon dioxide, it is forming carbamino compound, and it is also forming this bicarbonate ion inside the RBC. Now, what is the fate of this bicarbonate ion inside the RBC? So, whenever the bicarbonate ion is high inside the RBC, okay, it will enter into the plasma. It will enter into the plasma. Why? So, it will cross the membrane of the RBC. So, now the bicarbonate is reaching into the plasma. But this required the help of one exchanger molecule that will exchange chloride from the plasma. So, chloride molecule is reaching from plasma into the RBC, and in exchange bicarbonate is going into the plasma. So, this carrier protein or this transporter is known as the anion exchanger protein or band 3 protein. This is known as the band 3 or anion exchanger 1 protein. Getting my point? So, because of the presence of this anion exchanger, the bicarbonate is extruded out of the RBC and chloride is entering inside the RBC. So, this is chloride bicarbonate antiporter, which is also known as anion exchanger or band 3 protein. And this phenomenon by which chloride is exchanged into the from the plasma to the RBC, this is known as the CHLORID, chloride shift phenomena. Chloride shift phenomena is also known as Hamburger effect. Okay, Hamburger effect, right? So the chloride shift or Hamburger effect, this effect you have to remember for your purpose of MCQ. So the chloride shift or Hamburger effect, whatever. Okay. Now, so bicarbonate ion is produced inside the RBC. So either the bicarbonate will be high, or if bicarbonate is exchanged with chloride, then the chloride ion will be high, because this much amount of chloride was not there inside the RBC, but this is coming because of the exchange with bicarbonate. So whenever this chloride content is high inside the RBC, it will increase the osmotic tension of the RBC. It will increase the osmotic tension. So osmotic tension of the RBC will be increased. So I will write down here that there will be increase in osmotic tension of the RBC. And whenever osmotic tension increases, what happens? The RBC will drag some water from the plasma, means water will enter inside the RBC. And whenever water is entering inside the RBC, what will happen to the size of the RBC or the volume of the RBC? Because of this water entry, the individual size of the RBC will be higher. And because the individual size is higher, if you take the packed cell volume, that is the volume of the total RBC, this will be 3% higher in venous blood, 3% higher in venous blood. Again, I am repeating. So because of the increase in the chloride content inside the RBC, the osmotic tension of the RBC will be higher, because osmotic tension depends on the concentration of the molecule and the number of molecule. So whenever the osmotic tension is high, it will drag water, that is the osmosis. So because of the dragging of water from plasma inside the RBC, the volume of the RBC will be increased a little bit. And when the individual volume of the RBC is increased, what will happen to the total volume of the all RBC, that is known as the packed cell volume? It will be 3% higher. Higher in which blood? That is the venous blood, because whenever the carbon dioxide is entering inside the RBC, this is which blood? This is your venous blood. That's why we say the blood, venous blood, the PCV is 3% higher than that of the arterial blood. Okay. So now we learn what is Bohr effect and what is chloride shift and how the carbon dioxide is transported in the venous blood inside the RBC and inside the plasma. Now when this RBC will reach from the tissue to the level of lung. So this is the venous blood. Now the venous blood will flow to the level of lung. Okay. So now we'll look at what is happening at the level of lung. So whenever this venous blood is coming inside the lung, we all know that the hemoglobin which is present in the venous blood or the RBC, venous blood RBC, that is the deoxyhemoglobin, that is HHB, that is the deoxyhemoglobin. So now this deoxyhemoglobinated blood is reaching at the level of your lung. So this is my representation of the lungs. So lungs contain huge amount of oxygen because we are taking continuous fresh air inside the lung. So this oxygen, first it will enter inside the plasma and further it will enter inside the RBC. Now the oxygen has entered inside the RBC. Now what is the fate of this oxygen? So obviously this oxygen is going to react with the deoxygenated hemoglobin. Simple. So this oxygen is going to react with the deoxygenated hemoglobin, that is the HHB. And whenever it is reacting with the deoxygenated hemoglobin, it is going to form oxygenated hemoglobin again, HbO2. Now HbO2, it's a acidic molecule. It's an acidic molecule. Acidic molecule means it's a proton donator, means it will not be able to hold the H plus ion. So it will release the H plus ion immediately. So look at this reaction. So it was HHB plus O2. So combination of this is giving rise to oxygenated hemoglobin plus separation of the H plus ion. Now what is the fate of this separation ion? So this H plus ion will be buffered. So who is going to buffer this H plus ion? So this H plus ion will be buffered by the bicarbonate ion. Okay, where from this bicarbonate ion is coming? So we have seen that in the venous blood, bicarbonate was going out of the RBC. Now the bicarbonate will be taken inside the RBC by the same transporter, and the chloride which already entered inside the RBC in the venous blood, those chloride will again put back into the plasma. So the chloride is again going back into the plasma and bicarbonate is entering. Why the requirement of bicarbonate is there? Because now this bicarbonate is going to neutralize this H plus ion. So H plus ion and bicarbonate, that is going to form the carbonic acid. Now this carbonic acid is going to form the carbon dioxide, okay, carbon dioxide and H2O, okay. So I'll write down this is clearly. So it is going to form the carbon dioxide and H2O. Now this carbon dioxide, because it's a gaseous molecule, it will be released from the RBC, okay, it will enter into the plasma and from there it will enter into the lung. So this carbon dioxide is released at the level of lung, okay, and where from this carbon dioxide is coming? Look at the whole reaction. So lung is delivering oxygen inside the RBC. Because of the delivery of the oxygen, H plus ion is released, and that H plus ion is combining with the bicarbonate ion, because of which carbonic acid is formed. Carbonic acid is dividing into opposite direction, that is carbon dioxide and H2O. So this carbon dioxide, because it's a gaseous molecule which is produced inside the RBC, it will immediately enter into the plasma and from there it will be released at the level of lung. So at the level of lung, because the oxygen tension is very, very high, so this high oxygen is helping in release of carbon dioxide from hemoglobin. So basically this is release of H plus ion, but that H plus ion is ultimately forming the carbon dioxide. So we can say that in presence of huge oxygen, the carbon dioxide is released from hemoglobin, and this carbon dioxide release phenomena is known as Haldane effect. So the Haldane effect is dissociation of the carbon dioxide from hemoglobin, release of carbon dioxide from hemoglobin, release of carbon dioxide from blood into the lung when the oxygen is high. So whenever the hemoglobin is oxygenated, carbon dioxide is released from the hemoglobin or blood, and that is known as the Haldane effect. Getting my point? Haldane effect. So the chloride is exchanged in the plasma and bicarbonate is entering at the level of the lung. This is known as the reverse chloride shift. This is known as the reverse chloride shifting, right? And this same exchanger is doing this. And now you look at, so carbon dioxide is released, but excess water is still there. But because the osmotic tension of the RBC will decrease, because the chloride is going out of the RBC, so osmotic tension will decrease. So because of the decrease in the osmotic tension, the water which was released inside the RBC, this water is also going out of this RBC. So because of the excess water released from RBC, what will happen to the PCB? PCB will be normalized inside this. Okay, PCB will be normalized. So PCB normalized means this is which blood? This is your arterial blood. This is your arterial blood. Okay, getting my point? So whenever there is oxygenation of hemoglobin is happening at the level of lung, the blood is converted into arterial blood. But the blood which is coming here at the level of lung, we all know that this is the venous blood. So the venous blood is coming. So the venous blood is containing this deoxygenated hemoglobin that is become oxygenated, converted into arterial blood. Simultaneously it is releasing carbon dioxide, and this phenomenon is known as Haldane effect. So how the Haldane effect will be described? It is described in this way, that oxygenation of the blood in the lung displaces carbon dioxide from hemoglobin and increases the removal of this carbon dioxide, and this is my Haldane effect. The Haldane effect describes the shift in the carbon dioxide dissociation curve caused by oxygenation of the hemoglobin. So Haldane effect is basically related to the carbon dioxide dissociation curve. Please note down here, Haldane effect is not the left shift of the oxygen hemoglobin dissociation curve. Sometimes we describe like this, that Bohr effect is the right shift of the oxygen hemoglobin dissociation curve in presence of carbon dioxide or H plus ion, and Haldane effect is opposite, it is the left shift. No. Haldane effect is not related to the oxygen hemoglobin dissociation curve. Haldane effect is described in terms of carbon dioxide dissociation curve, which you have not learned yet. So there is shifting of the carbon dioxide dissociation curve, and that shifting of the carbon dioxide dissociation curve in the downward direction is known as Haldane effect. So what is this carbon dioxide dissociation curve? That we are going to understand now. So before that, Haldane effect, remember that Haldane effect approximately double the amount of carbon dioxide released from the hemoglobin in the lung, and it double the carbon dioxide pickup in the tissue. So whenever you are releasing carbon dioxide at the level of lung, what will happen? So arterial blood carbon dioxide will be lesser. So this arterial blood, whenever it will reach to the level of tissue, it will take up carbon dioxide from the tissue level. So this release of carbon dioxide because of the Haldane effect is double, and whenever the release of carbon dioxide is double from the tissue, you can take up double carbon dioxide, and that is written here, that because of the Haldane effect, the carbon dioxide release is double at the level of lung, and because of this Haldane effect, the pickup of the carbon dioxide is also double from the level of tissue. Whatever is released, that is taken up from the tissue. Okay, that is the thing. Now I will describe that how this carbon dioxide dissociation curve is described. Okay. So carbon dioxide dissociation curve, x-axis is the pCO2 and y-axis is the volume of carbon dioxide. You have to listen carefully, very carefully. Okay. So first of all, I am drawing the carbon dioxide dissociation curve. So this is the carbon dioxide dissociation curve. Okay. So this is the carbon dioxide dissociation curve of arterial blood curve. So whenever I am saying arterial blood curve, so what is the arterial blood partial pressure of oxygen? That is 100 millimeter of mercury. You all know that. So when the partial pressure of oxygen is 100 millimeter of mercury at the level of arterial blood, carbon dioxide dissociation curve is this is the black color curve. So in arterial blood, okay, what is the partial pressure of carbon dioxide? That you have to remember, that is 40 millimeter of mercury. And what is the partial pressure of carbon dioxide in the venous blood? That is 46 or 45. So when the partial pressure of carbon dioxide is 46 millimeter of mercury, okay, so this x-axis is the carbon dioxide. So whenever the partial pressure of carbon dioxide is 46 millimeter of mercury, okay, with this 46 millimeter of mercury point, I am drawing a perpendicular on this carbon dioxide dissociation curve. I am drawing on perpendicular here. Okay, so let me draw, okay, quickly. So this is the perpendicular on this carbon dioxide dissociation curve, and from there, if I drop another perpendicular on x-axis, that will be the volume of the carbon dioxide. So this volume of carbon dioxide is 52 ml. I'll explain what is this. So when the venous blood partial pressure of carbon dioxide is 46, with this partial pressure of carbon dioxide, how much ml of carbon dioxide is transported in the venous blood? 52 ml. 52 ml. Okay. Now here I'll make one correction. So this is the curve that is not the arterial blood curve. So this is the curve which is the venous blood curve, because everything we are discussing the venous blood. So venous blood carbon dioxide dissociation curve, it is here. So if it is venous blood carbon dioxide dissociation curve, what is the partial pressure of oxygen here? That is the 40 millimeter of mercury. Okay. So in venous blood, when the partial pressure of oxygen is 40 millimeter of mercury, the partial pressure of carbon dioxide is how much? 46. When the partial pressure of carbon dioxide is 46, how much is the amount of carbon dioxide present in the venous blood? That is 52 ml. Okay. Now, whenever this blood will be converted into venous to arterial blood, what will happen to this partial pressure of oxygen? Partial pressure of oxygen in the arterial blood is 100 millimeter of mercury. But note down, when it is arterial blood, this carbon dioxide dissociation curve will be shifted in a downward direction. So now we look at, so this blue color curve, this is also carbon dioxide dissociation curve, but this is the carbon dioxide dissociation curve of the arterial blood. Getting my point? Okay. So in arterial blood, what is the partial pressure of oxygen? The partial pressure of oxygen is 100. Okay. 100. Now we look at, so whenever the venous blood partial pressure of oxygen which is 40, whenever this partial pressure of oxygen is raised to 100 millimeter of mercury, the carbon dioxide dissociation curve is shifting in the downward direction. The carbon dioxide dissociation curve is shifting in the downward direction. So in the arterial blood, now you try to understand that in the arterial blood partial pressure of oxygen is 100, but how much is the partial pressure of carbon dioxide? So in the arterial blood, partial pressure of carbon dioxide is 40. So this is the arterial blood carbon dioxide, and previously we have discussed the venous blood phenomenon. Okay. So in arterial blood partial pressure of carbon dioxide is 40. So from here, if I drop on perpendicular on this arterial blood curve, so I am drawing on perpendicular from here on the arterial blood curve. Okay. And that perpendicular I am extending on the y-axis. Then I will find out the carbon dioxide content in the arterial blood. Okay. And this value will be 48 ml. So now we look at, so when the partial pressure of carbon dioxide is 40, that is in the arterial blood, how much carbon dioxide is present? 48. So what is the difference between these two? That is 48 to 50, that is 4 ml. So look at, because of the oxygenation of the hemoglobin, because of the increase in partial pressure of oxygen, what is happening? The carbon dioxide dissociation curve is shifting in the downward direction. And this downward shifting of the curve, this downward shifting of the carbon dioxide dissociation curve, okay, carbon dioxide dissociation curve is known as Haldane effect. So how the Haldane effect is helping in carbon dioxide transport? Okay, so suppose there is no shifting of the curve. Suppose this curve is not shifting in the downward direction, means Haldane effect is not there. Then what will happen? Then when the partial pressure of carbon dioxide is 46 in the venous blood, the content of carbon dioxide is 52. But when the partial pressure of carbon dioxide is 40, in that cases, how much carbon dioxide will be transported by the blood? Look at here. So from this 40, the curve has not been shifted. Because the curve had not been shifted, I am dropping on perpendicular from this 40 point onto the original curve, that is the black color curve, because there is no shifting. So from there, if I drop another perpendicular on this axis, y-axis, so I can easily find out here that if the curve is not shifting in the downward direction, then venous blood carbon dioxide partial pressure is 46 and arterial blood partial pressure carbon dioxide is 40. What is the difference of content of the carbon dioxide between these 2 points? So this is 52 and here it is 50, means the difference between these two is only 2 ml. So if there is no shifting of the curve in the downward direction, then the difference of carbon dioxide between this venous blood and arterial blood is how much? That is only 2 ml difference. But because the curve is shifting in the downward direction, the difference between the venous blood carbon dioxide and the arterial blood carbon dioxide is how much? That is the 4 ml. So the Haldane effect is basically doubling the carbon dioxide transport from 2 ml to 4 ml. So that's why we say in the previous discussion that Haldane effect approximately double the amount of carbon dioxide released from the hemoglobin in the lung, and same amount of carbon dioxide is picked up from the tissue. Getting my point? So if you look at the Book's diagram of this Haldane effect or carbon dioxide dissociation curve, so this is the curve in the venous blood. So this is the curve in the venous blood. So venous blood means partial pressure of oxygen here will be 40 millimeter of mercury. And whenever you are causing oxygenation of this hemoglobin, it will be converted into arterial blood. So whenever the partial pressure of oxygen is 100, the curve is shifting in the downward direction, and this downward direction shifting is known as the Haldane effect. And because of this Haldane effect, we can find out that this carbon dioxide content in the venous blood and the carbon dioxide content in the arterial blood, the difference between them is 4 ml. Otherwise, if there is no shifting, the difference will be only 2 ml. And that's why we say that Haldane effect double the carbon dioxide transport. Okay. So now we are going to discuss certain MCQ from this topic. So the carbon dioxide is primarily transported in the arterial blood or venous blood, whatever you say. The main is the bicarbonate, that is approximately 70% of the carbon dioxide is transported in bicarbonate. And the next, increase oxygen delivery to the tissue in response to increased carbon dioxide. So in presence of carbon dioxide, oxygen is released from the hemoglobin, and that is nothing but your Bohr effect. So this is all about carbon dioxide transport.